Good morning. Good to see you there. Alessandra, I should say. Are you able to share your screen for the first presentation today?
Yeah.
Great. I can hear you just fine. And I can see your screen coming up. That's great. Thanks everybody for coming in today and looking forward to learning about some more kinases today. And you can start whenever you're ready, Alessandra.
You can see it. All good.
Yeah, it's perfect.
Okay. Okay. Hi, everyone. Today I'm going to be talking about MAP kinase interacting serine threonine protein kinase 1, which is also known as MNK1. This protein is found coded on chromosome 1 in humans. It is contained in all human tissues with the highest expression in the blood, with also great ubiquitous expression in the kidneys, liver, pancreas, and many more. It is localized to the nucleoplasm. However, it may also be contained in the cytoplasm, and the other two isoforms that are generated by alternative splicing are found in both the cytoplasm and the nucleus. In terms of the function, it responds to environmental stress and cytokines through the activation of P38 mitogen activated protein kinase. And this kinase, along with ERK, or extracellular signal regulated kinase, go on to phosphorylate MNK1, activating it. And once MNK1 is active, it goes on to phosphorylate eukaryotic initiation factor 4E through an interaction with the C-terminal region of EIF4G. And once EIF4E is phosphorylated, its affinity for the 5' mRNA cap greatly increases, leading to cap-dependent translation to proceed. And this is evident through mutations in MNK1 and 2 that resulted in diminished cap-dependent translation. The other functions are not known as well. In terms of the structure, there's only one domain, which is the kinase domain, that lies within residues 49 and 374. The kinase domain exhibits strong sequence similarity to other kinase domains in proteins belonging to the calcium calmodulin dependent protein kinase family. The protein does contain a nuclear localization signal near the N-terminus, which consists of basic residues allowing its entry into the nucleus. And it does contain some disordered regions near the N and C termini. And here, this is just the catalytic domain, not the entire protein. For interactions and regulation, the protein does not appear to be lipid modified. There are 3 key post-translational sites contained at threonine 250, 255, and 385. These are done by the P38 MAP kinase as well as ERK, and they're required for activation of the kinase domain. Looking at the lollipop plot, you can see that there's 2 ubiquitination sites as well as 2 methylation sites shown in gray. However, these are not in regions where the protein is predicted to possibly interact with membranes. However, there are other regions where the protein may interact with membranes. In terms of mutations and disease, it's not a lot of data about one specific cancer. However, it may be a potential marker for hepatocellular carcinomas, as well as deletions have been associated with chromophobe renal cell carcinomas. What is known is that missense substitutions are the most frequent type of mutation present in cancer samples. And interestingly, adenovirus progression may result from a viral protein docking onto EIF4G, causing displacement of MNK1, which leads to the absence of EIF4E phosphorylation and halting translation. And I would expect that this protein is overexpressed or highly mutated in cancer, considering its relation to translation, as well as its potential link to the cell cycle through the MAP kinase and ERK pathway. And here's my references.
Great. There's some questions appearing in the chat space. Can you answer those?
Yeah. Have there been any drugs developed to target the kinase? When I was looking through the databases in Cosmic, it didn't appear that there were any specific types of drugs. I think especially because there's not too much cancer research and like any specific tissue cancer research. So, yeah.
Maybe one more question.
Okay. Can you tell me more about the interaction between MNK1 with the C term of EIF4G? Yeah, so EIF4G acts as like a scaffold protein where MNK1 and EIF4E both bind to it. And this allows for MNK1 to phosphorylate EIF4E just through like the vicinity or being close to one another. So in the absence of EIF4G, the proteins are not able to make contact with one another.
Okay, great. I want to thank you very much for a great first talk and move on to the second just to stay on schedule if we could. Justin, can you share your screen?
Yes.
Okay. Thank you. It's just coming up now. Feel free to start as soon as it displays in a second. Okay, you're good to go.
Did it display?
Yep, you're good to go.
Perfect. All right. Hi, everyone. I'm Justin, and today I'm going to talk a little bit about a member of the dark kinome called eukaryotic elongation factor 2 kinase, also known as EF2K. So, eukaryotic elongation factor 2 kinase is a globular serine threonine kinase with mainly alpha helical structure. It has a single alpha type kinase domain, which is shown on this figure here in red. And it is also dependent on the calcium calmodulin complex for signaling, and that binds at the yellow residues here. It also depends on ATP binding for its activity, and that's shown with the orange. It has only one definitively known substrate called eukaryotic elongation factor 2. And Phoebus and Phileus do not predict any transmembrane regions or signal peptides, indicating it's a globular protein. So by now you may be wondering, what does this kinase do and where does it operate? So really, EEF2K's main function is to regulate translation in the cell. Its substrate EEF2 binds to the ribosome and is required for peptide chain elongation during translation. Phosphorylation of EEF2 by EEF2K releases it from the ribosome, and this stops translation. The kinase functions in cells in all tissues, as translation is needed in all tissues, but it's predominant in skin and brain tissue. It's primarily localized to the cytoplasm where translation occurs, but can also be found in the nucleoplasm. However, how it gets there and under what conditions it localizes to the nucleus are not yet known. So this could potentially be a control mechanism, maybe. So as far as the regulation is pretty complicated and it's not very well understood, but it is regulated by phosphorylation by a variety of kinases. In particular, low energy signals like AMP can activate AMP kinase, which will phosphorylate and activate EEF2K, which will then inhibit translation. On the other hand, cell proliferation kinases such as MAP kinases or CDKs phosphorylate and inhibit EEF2K, which will ultimately activate translation. EEF2K is also known to be autophosphorylated on at least 9 residues, although whether these phosphorylations are stimulatory or inhibitory depends on the site. So what's interesting here is that all these phosphorylations, apart from whether they're inhibitory or stimulatory, nothing is known about the mechanisms. So to look into this a little bit more, I started looking at membrane and lipid interactions of EEF2K and post-translational modifications. So D2P2 does predict a disordered protein region that may bind lipids between residues 300 to 500. And the disolipid results here in the bottom panel mirror that as well. And notice that the phosphosite results show a lot of phosphorylations in this region, which could be potential PIP stops. MODA is the middle panel. It also predicts a number of possible membrane or lipid binding sites. And in these regions, there are 7 phosphorylation sites and 7 ubiquitination sites. So these could be PIP stops as well that allow EEF2K to dissociate from a membrane and localize to the ribosomes to regulate translation. One thing I also wanted to note is there is a predicted palmitylation at cis 212 by Swiss palm, although it has never been observed experimentally. So for the biomedical significance, overexpression of EEF2K is associated with some cancers, particularly prostate cancer and leukemia, although there's a number of other cancers as shown with this expression plot here. Missense mutations are also associated with cancer, and these are located all over the protein, not just the kinase domain. For example, there's a lysine 347 that is mutated to an asparagine in a patient with uterine cancer, and this is located in the motor membrane binding region. So as a result of its implications in cancer, EF2K remains a hot topic of biomedical research and probably will for many years to come. So with that, here are my references, and I'll be happy to take any questions in the chat or right now.
Great, Justin. Maybe you can answer the first question in the chat there.
Sure.
Where are the autophosphorylation sites in relation to the 3 domains discussed?
Yeah, so of the 9 autophosphorylation sites, most of them are in the disordered regions of the protein. So there aren't any autophosphorylation sites in the kinase domain or in the calmodulin or ATP binding sites, but they're mostly in that 300 to 500 disordered region.
Great, Justin. Maybe you can answer the other questions in the chat space. We're just running a little tight on time.
Yeah,
and
for sure.
Kaitlin, could you share your screen? Great job, Justin. Thanks for all the speakers so far this morning. And Kaitlin, we can see you, and I think your screen's coming up. So as soon as it's there in a second, you can go for it now, maybe just full screen. Perfect. You're good to go. Just unmute.
So today I'm going to be talking about the tumor-promoting 53 regulating kinase. It's an atypical serine-threonine kinase. So it's on the Dark Kinase website. It indicates that it's part of the CK1 family that commonly phosphorylate key regulatory molecules that are involved in cell cycle and play essential roles in tumor progression. It's applied to TP53RK as it phosphorylates downstream P53, causing upregulation of the inhibition of cell proliferation, resulting in apoptosis. It's component of the kinase, endopeptidase, and other proteins of small size, so KEOPS, and endopeptidase-like and kinase associated to transcribe chromatin EKC complex. It's required for the formation groups in tRNAs that read codons beginning with adenine, and kinase ATPase activity is theorized to support the catalytic subunit of the complex. It's part of the BUD32 kinase family, and that is the kinase subunit in the complex. It's expressed broadly throughout the body, but it's highly in the testes, and it's suspected to be localized in the cytoplasm and nucleus. The structure in uniprot indicates that there's a nuclear localization signal motif within the kinase domain from residue 78 to 95. Due to it being on the exterior alpha helix and densely rich in arginine, it's indicated that there may be true signals, but there's no information currently on the nuclear transportation method of the kinase. There's 2 more domains. There's a disordered region from residues 1 to 21 and a kinase domain that spans from 22 to 253. In vitro, it's a protein kinase, but when associating with the KEOPS complex, the catalytic subunit OSGEP switches the activity from a kinase into an ATPase. There's 2 ATP binding sites on the residue from 39 to 47 and at residue 60, and there's a proton activation site on residue 162. Phobias indicates that there is no transmembrane domain as the signals were not over the required scores. This is consistent with the overall amino acid makeup as the kinase isn't overly hydrophobic, showing it's likely not inserted in the membrane. MODA confirmed that there's no membrane binding sequences as there's only 1 score over 30 and on the C-terminus end, but it's likely due to positively charged amino acid, so it's a false signal. Desolipid indicated a disordered lipid binding region at the C and N terminus, but it's unlikely as the membrane binding is likely due to charged and disordered ends of the kinase, and there's also no coiled coils. So there are 7 phosphorylation sites on tyrosine, serine, and threonine residues throughout the kinase. AKT is known to phosphorylate serine 250, which is important in the activation of the kinase. And these phosphorylations could indicate different lipid binding sites, but it's unlikely due to no transmembrane region. It could play a role in regulation and activation or inactivation of the domain. And phosphorylation at site 34 is within 5 residues of an ATP binding site, indicating it has a function in the activity and the kinase as a whole. There's 3 ubiquitination sites on lysine residues throughout the kinase. It could be altering the kinase activity when it's in the complex. One ubiquitination site is within the ATP binding region, meaning it along with the phosphorylation site nearby causes the activation of the kinase may be with the phosphorylation site. There are 2 methylation sites on arginine residues 68 and 75. Methylated arginines have been shown to influence the DNA damage response, which would be likely in this situation as the kinase dysregulation has direct effects on p53 and tRNA modification. So the health implications, most frequently occurring cancers are colorectal and endometrial cancers due to amplifications in the red, and the green shows the mutations. TP53RK inhibition results in the inhibition of p53, causing cell survival and proliferation. There's point mutations on residue 42 from a G to a D and residue 81 from a T to a R. The changes eliminate interactions with the kinase binding protein, resulting in Galloway-Mowat syndrome, which is a severe renal neurological disease with many developmental delays and seizures. There's no treatments or symptoms for it, and symptoms are managed. So those are my references and questions.
Great. Let me just see if there's any questions here. Allison is asking, how can gamma symptoms be managed?
So for each patient, it's kind of different. They can present with more severe symptoms, but it really depends on the patient. So some can have real severe kidney issues, so it can be treated with dialysis or medication. And then some have really bad seizures, which can be treated with anti-seizure medications as well.
Yeah, I just have a quick question as well. Did the nuclear localization motif overlap with that weak MODA signature?
I'll just go back if it lets me.
Because they're both basic elements, right? So I've been
Yeah,
thinking about that. If it's false positive on a nuclear localization,
it kind of is on the edge of it because the nuclear localization signal is from what point is it from? 78 to 95, and the MODA signal is right on the edge of like 72. So it kind of does, but it not really.
okay,
So it's hard
fascinating.
to determine.
Thanks so much, Kaitlin. You've done a great job. Maybe we can ask you to answer the other questions in the chat. And then, Kevin, are you able to share your screen? Great. We can see your slides and maybe just full screen, and you're good to go.
Perfect. So hi, everyone. Today I'll be talking about ALPK3 kinase for my presentation. ALPK3 stands for alpha protein kinase 3. Its unipod enzyme class are listed on the slide here. Its expression level is sort of broad, but it's concentrated primarily in the heart as well as in the skeletal muscle.
Kevin, we don't see the full screen yet, so can you double click on the present again one more time? Oh, yeah, that's better. We can see, yeah, just I can see your second slide with the expressions. So yeah, that's fine. Maybe just click through on the bottom might be easier.
Okay, like this. Okay.
Yep, perfect.
Okay, so the subcellular location is in the nuclear envelope, specifically in cardiomyocytes, as well as the M band of the sarcomere, where the sarcomere is a fundamental repeating unit of muscle, specifically skeletal muscle. In terms of the structure of ALPK3, it's composed of 1,907 amino acids and contains 3 different domains, an alpha-type protein kinase domain, as well as 2 Ig-like domains. It's a non-cytoplasmic protein, and there are no predicted signal peptide sequences or transmembrane helices. In terms of lipid modifications, there are no palmitoylation, no misrelation, no farnesylation, and no geranyl as well. ALPK3 has one pip stop located at serine 1424. And the way I was able to deduce this was that for all alpha kinases, there is an invariant lysine residue located at the position 1420. And since this phosphorylation site is within 5 residues, it likely functions as a pip stop. ALPK3 has limited kinase activity. So what I mean by that is some studies, specifically by Agarwal et al., had described ALPK3 as a pseudokinase. So a pseudokinase is a pseudovariant of an enzyme that lacks catalytic activity. However, some other studies have demonstrated that ALPK3 phosphorylates various proteins, including SQSTM1. So I sort of found the middle ground and likely deduced that ALPK3 functions as a weakly active kinase. And if we look at the sequence alignment of the alpha kinase family, it sort of supports my conclusion. So the red represents entirely conserved residues among all members of the alpha kinase family, whereas the green represents conserved among all members except for my protein, ALPK3. And what we can see here in this schematic is that all of those non-conserved residues are located within the interlope cleft. And these are sort of involved in binding to substrate. So two of them are involved in positioning ATP, and one of them is involved in substrate binding. And so the lack of those conserved residues likely means that ALPK3 has weaker kinase activity compared to its counterparts. In terms of function, ALPK3 is critical for sarcomere and cardiac function. So it's a myogenic kinase located at the end band of the sarcomere. It's involved in sarcomere organization as well as calcium handling. So in loss of function ALPK3 mutant mice, they show an elevated level of calcium in their hearts, and that sort of mirrors arrhythmias that we see in patients. ALPK3 also maintains cardiac contractility. So what we can see here in this graph is that in ALPK3 mutant mice, they have a lower contractility compared to its wild-type counterparts. ALPK3 is required for the sarcomeric localization of SQSTM1, and it's critical for the phosphorylation of sarcomeric and protein quality control proteins. And we can see this in this volcano plot over here, where ALPK3 loss of function mice have significantly decreased levels of phosphopeptides compared to its wild-type counterparts. And so ALPK3 variants overall cause myomucin accumulation and disrupted cardiomyocyte sarcomere structure in pathology. So the levels of both myomucin 1 and myomucin 2 were increased, as were the levels of thick filament proteins. So by having an increased level of these proteins, it causes a higher protein density, and this can decrease contractility and make it sort of evolve into hypertrophy in patients. So this could be summarized in the model over here. So in wild-type mice, ALPK3 and myomucins lead to force buffering at both the nuclear envelope as well as the sarcomere end band, and it helps mediate protein turnover. However, in loss of function models, the ALPK3 and myomucins are disrupted, and this could lead to impaired nuclear force buffering as well as impaired end band protein turnover. And this culminates into dilation, hypertrophy, as well as depressed function overall. Thank you.
Okay, great. There's a question from Takdir. Is there a way to combat the low contractility rate caused by ALPK3?
Yeah, thank you for that question. So currently, the clinical side associated with the decreased contractility have not been sort of like there hasn't been no sort of drugs or any sort of methods used to combat it, but I hope in the future it could be elucidated.
Great, Kevin. That was a great talk. If there's any more questions, just answer them in the chat. I'd like to invite Natalie to share her screen. You guys are all doing a great job, and we're just kind of motoring through, and lots of good questions in the chat space. Natalie, your screen is coming up. That's great. Also unmute. And can't quite see it yet. Oh, there we can see it. If you can full screen, that'd be fantastic.
Okay, there we go. Sorry, just getting it. Yep.
Perfect. It's coming up, just loading.
Okay, everyone can see it
Go
now.
for it.
Perfect. So for my project, I chose the kinase BRISC2, which is a serine threonine kinase. Over here, I have kind of the AlphaFold predicted model here, which, as you can see, it basically just decreases in confidence as you go out from the core of the protein, which makes a lot of sense because the regions kind of get more flexible. I actually wanted to talk about some of the issues that I ran into while I was researching my kinase. So first of all, of course, it is one of the dark kinases. So all of the sources are super, super vague, a lot of times just pure speculation, and often even contradicted each other. A big example I had of that was, as you can see, I had a phobias prediction here, like really, really strongly that there was a transmembrane domain. Yet other sources like BioLib, which I have the result of down here, had absolutely no indication of this. So while in this case, it turned out to be this cyan helix here causing, I believe, a false positive on the Phobias website. It was kind of hard to tell what I was dealing with when it came to some of the other websites. And then as well, the human form of BRISC2 itself doesn't actually have its own PDB entry. Well, there were 2 mouse forms that had over 90% sequence identity. As you can see here, so the dark blue one here is the human one, and the red and green are the 2 different mouse PDB forms that had their own entries. They don't actually overlap that well, so they didn't end up being as good of like estimations as I hoped they would be of my protein structure. So with that being said, the function, of course, is, as I mentioned, it's a serine threonine kinase. It seems to be involved with neuron polarization, the cell cycle, as well as insulin secretion. Within the cell, it is found in the perinuclear cytoplasm. So that's like the light blue circle here on the, this is like the little cell diagram that UniProt gives you. And then in terms of tissues, it's found mostly in the brain and the pancreas. In terms of domains, it's got this protein kinase domain here in blue. Inside the protein kinase domain, there are 2 ATP binding sites as well as a proton accepting active site. This orange one here is a ubiquitin binding domain, and it's very near this Ken box motif. The Ken box motif is necessary to bind CDH1 for CDH1 mediated ubiquitination. So it makes sense that those are right next to each other. And then as well, this protein has 2 compositional biases. This one here is acid-base compositional bias, and then up here in yellow, there is a proline compositional bias. And then of course, there's a lot of disordered regions in this protein as well. Post-translational modifications, there seems to be some methylation in the middle here. Again, as you can see, most of these are not very well researched. There's a ubiquitination site thought to be on either end of the protein. There's also a sumylation suggested somewhere in the middle of the protein, but most commonly, as you can tell from all of the blue dots here on the phosphostate diagram, it's mostly phosphorylations. For membrane binding, this was where I tried to use MODA and it did not work, and the most ones were not good approximations, but thankfully the PMI-Pred website gave me this really nice model. So as you can see, that proline compositional bias that I outlined earlier was a strong contender for a membrane binding region, and it's thought to be surrounded by the by like some membrane sensing regions. This would explain why the most ones did not have very good results on MODA, because they don't have these loop regions like the human form of BRISC2 does. And then as well, this is really important information because one of the suspected interactions that BRISC2 has is with the ER membrane during its stress response. And then finally, the mutagenesis invariants. Basically, all of the mutations of this protein either completely shut it off or completely turn it on. And there's over 800 variations, which you can see. This is the figure from UniProt here that it gives you for the variants. The variants have a very, very wide range of impacts, ranging from pretty much no impact, but a lot of the more severe ones are intellectual disabilities as part of what's called BRISC2 related disorder. So yeah, and then these are my sources.
Great. That was very impressive. You did a lot of digging there, and thank you for highlighting both the ambiguity and what you found. There's a couple of questions in the chat space. Does the proline-rich region have a function?
I couldn't find one like outlined, but like I said, it was on PMI-Pred there, it was suggested to be a membrane binding region, which would make a lot of sense because it would be very hydrophobic. So I do believe that that's probably a pretty good prediction, is that it's important for membrane binding.
Oh, okay. And are there any mutations linked to specific diseases?
I would have to just take a closer look at that in like the UniProt website and the different sources and stuff. Mostly what I was getting as results were just very vaguely like intellectual diseases. Some of the more recent research has been looking specifically into whether there's a connection to something like autism, which doesn't seem to be going that well, but overall, it just kind of seems to have general delays. Like I said, it's kind of all characterized under BRISC2 related disorder, or BRISC2 related disease, which is kind of just generally there are some intellectual disabilities as a result of the mutations.
Thanks, Natalie. That was very clear and excellent responses. Nicol, could you share your screen next, please? Is Nicol in the room? Maybe
Hello,
Nicol.
yes. Sorry,
you
one
are.
second. Let me just
I can
pull it
hear
up.
you, and yeah,
Let me just
here. Lots of questions there in the chat space for Natalie, so feel free to keep answering those in the chat space. I can see your screen coming up, Nicole. It looks great. Just waiting a second here more. Okay, I can see the screen. Maybe just full screen it if
Can
that
you
works.
see it now?
No, I just see your presenter mode. For some reason, this program isn't in Canva, but you can just click through those slides at the bottom.
Oh,
Wishes as well.
okay. Sorry about that.
I'm not quite sure why that doesn't work as
Oh,
well,
sorry.
but I can see your presenter view. The slides are just a little bit
Okay.
smaller than
Sorry
regular
about that.
size, but it's okay. We can probably see what's going on.
Okay. Can you see my screen?
Yeah, I can see your screen. And just when you click on present, I don't see your full
Okay.
screen come up. So, and I don't know if you have
Can
two
you see it
screens.
now?
No. Oh, wait a minute.
Oh, sorry.
Yeah. Yeah, that's great. Just click through the bottom of your screen.
Okay.
Like
I'll just have it. Okay.
Yeah, that's fine. We can see what's going on.
Okay. Okay. I'll just have it like this then.
Yeah, go for it.
Okay. So hi everyone. The kinase I chose was coenzyme Q8B, which is also known as COQ8B, and its alternate names are ADCK4 or ARF domain containing kinase 4. Sorry. So coenzyme Q8B is involved in the biosynthesis of coenzyme 10, and coenzyme 10 is more commonly known as ubiquinone, which is important for mitochondrial oxidative phosphorylation, and it's also an antioxidant in cell membranes for reactive oxidative species. So COQ8B is a mitochondrial protein, and it's expressed widely in a wide range of tissues shown here. But it is an atypical kinase. And what I mean by that, it was while it was initially proposed to be a protein kinase, coenzyme Q8B is part of the protein kinase-like family because it contains a conserved protein kinase domain, but it doesn't perform the typical mechanism by which protein kinases will transfer a phosphate group from ATP to a substrate protein. Instead, coenzyme Q8B exhibits ATPase activity by hydrolyzing ATP into ADP in the presence of coenzyme Q intermediates to enable the formation of this mature coenzyme Q. And so, sorry, I had animations with this, which is why
It's okay,
like
Nicole.
this.
I trust you on that.
I'll just move it. But I had while there is no published structure that exists for coenzyme Q8B, it does have an alpha fold structure shown here. And it has a single transmembrane spanning protein. And then this is the Phobius and Filius prediction. As you can see, they're quite similar, where it has just the single transmembrane protein domain. So the transmembrane domain is in purple, and then there's a protein kinase-like domain, which is the blue. And despite the sequence similarities to protein kinases, experiments that attempted to demonstrate the canonical protein kinase activity for these proteins for in vitro have been largely unsuccessful, and there is no direct evidence for phosphorylation of these proteins. It also has a KXGQ domain, which blocks a crucial part of what would be the active site. So it's the green here. So this is where like the substrate binding pocket would be, but because it has the domain, it's unable to perform phosphorylation. And because of this domain, it's thought to have an auto-inhibitory mechanism that prevents the typical protein kinase activity. There is other research that suggests that coenzyme Q8B can perform as a kinase for phosphorylating coenzyme Q3, which is another component of the coenzyme Q metabolon, but further research is needed to fully understand the catalytic properties for coenzyme 8QB. So because there is an exclusion in the substrate binding pocket, it doesn't necessarily mean that ATP cannot bind to its own nucleotide binding pocket. And so here it has ATP binding sites in this region. So it predicts that it has the ATPase activity and that ATP is the ligand and it interacts with the coenzyme Q intermediates. So, but further research is still needed to fully understand the catalytic properties of this. So then this is the phosphosite plus. It is showing that there could potentially be a ubiquitination site in terms of post-translational modifications. But again, this kinase isn't really as well studied. So yeah, then. So in terms of like clinical implications and of this, it has documented that there are mutations that cause a kidney disease called steroid-resistant nephrotic syndrome. And because coenzyme Q10 is affected, that means that the kidneys can't filter blood properly, and so it leads to high levels of protein leaking into the urine. And so then treatments for this disease include high doses of oral supplementation of coenzyme Q10 as a possible therapeutic strategy. Another disease that was published just recently in 2024 is that this gene can cause an eye disease called non-syndromic retinitis pigmentosa, which causes progressive blindness, which means that there's not enough coenzyme Q10 in the retina, so that it causes the photoreceptors to die, resulting in this disease. And then these are my references. Okay, any questions?
Great. Any questions for Nicol? Thank you for your work on the animations. You did a lot of work, so really appreciate that. Let me see if there are any questions coming up. Could you just remind me again about the that transmembrane domain?
Yes.
Is it partially inside the kinase structure, or is it pretty free, do you think, to engage the membrane?
It's not like, I wouldn't say it's directly there. It's like sort of like higher, a little bit higher up
Yeah.
than the protein kinase domain.
Yeah, remember the membrane is about 35 angstroms in depth typically. It can be plus or minus a bit, but about how long do you think that helix is that's exposed?
Sorry, I'd have to.
It's a bit of a tricky question, but I think it looks really interesting. It's certainly a possibility, but it's kind of borderline.
Yeah.
Any residues near that transmembrane helix that look like they could be membrane interactive?
When I put it through, I also was getting like very variable data, so none that I can like confirm. But I'd have to like look through it.
And Alessandra asks, are there any studies investigating the role that this protein may have on reactive oxygen species and their toxicity?
Not specifically because like this protein isn't that well studied, but because it is part of the biosynthesis of ubiquitinone, it would affect that pathway since it is like part creating it. So I guess you could target this this gene to create more ubiquitinone to then target reactive oxygenative species in a way.
Great. Thanks so much, Nicole. That's great. There may be a couple of more questions. Feel free to answer those in the chat if they appear. And our next presenter is Nidhi. Sorry if I'm mispronouncing your name.
No worries, it's actually Nidhi.
Okay. Well, thank you for clarifying. And thank you for presenting next. We can hear you and just share your screen there. That looks like it's coming up, and that's perfect. Great. Go for it.
Okay, so I'll start now. My name is Nidhi Patel, and I'll be presenting on the kinase SBQ1, which is also known as the SH3 domain binding kinase 1. And then here I have the uniport entry. It is a serine threonine protein kinase, and it is about 424 amino acids long, and the molecular weight mass is 46 kilodaltons. As for its primary expression, it is primarily expressed in the brain, but it is also present in the prostate, adrenal glands, and skeletal muscle at low levels. as shown on this image here on the right. And for subcellular localization, it is present in the cytoplasm majorly, but there are some predictions of it being present in the nucleus as well. Now for its function, as it is a serine threonine protein kinase, it is involved in the phosphorylation process and the cell regulation. And it is also involved in the signal transduction pathways that control the brain development. And this is through transmitting signals from like the cell surface near the cell surface to the interior of the cell. And it is also involved in cell proliferation. It promotes the ERK pathways, as shown here, as it binds to the SH3 domains of the proteins and allows for cell proliferation to occur. Now, regarding its structure, it does have the kinase domain from residues 53 to 318, and it shows that it has two low complexity domains. It is disordered as shown here on the IUP PRED3 database, and it is near towards the N terminus and the C terminus. As BK1 does not have a transmembrane region, as shown on the DIPT-MM database, and it does not have any coiled coils either. As for its post-translation modification, Swiss Palm was able to predict the palmotylation sites on cysteine residues 5, 16, and 17. And it also has the phosphorylated sites as shown here from the Phosphosite Plus database. Now, regarding its mutations, most of the mutations are related to the brain as that is where it's mostly expressed. So most of the mutations are from either pituitary gland tumors or cancers. And one of the ones that's more prominent is the spindle cell oncocytoma. And the mutation is a frame deletion from a glycine at 304 residue. And it leads to the activation of the MAPK pathway. And this leads to uncontrolled cell proliferation. And the primary treatment for this is the transsphenoidal surgical resection and also radiotherapy. However, sometimes it's not able to get rid of everything at the same time. So there have been recurrences where it comes back. So there have been trials ongoing pharmacologically to inhibit the MAPK pathways. And there are over 400 variants linked to SPK1 as shown on the graph on the right. And most of these mutations do lead to cancer, so they are carcinogenic. And most of them are related to the brain, as I mentioned, and some are present in the lung or ovarian cancers or cervical cancers as well. And this is because it affects the MAPK pathways and it inhibits apoptosis as well as causing uncontrolled cell proliferation. Okay, these are my references, and thank you.
Thanks very much. That was great. Any questions in the chat space, feel free to ask there. In the meantime, I'm going to ask the palmitylation sites. You found 3 of them. That's great. But on the other hand, it's mainly cytosolic. I'm wondering if you can reconcile that and if there is any other membrane binding features in the vicinity of that N-terminal palmitylation motif.
I was not able to find much on it, but I could take a look and add it onto the chat here and see if I can find anything. But as far as the transmembrane domains or anything, there was none present or any membrane binding. It was mostly present in the cytosol and it showed expressions of it just passing through the nuclear pores to go to the nucleus for translation.
Oh, okay. Was there a nuclear localization signal?
Unfortunately, I was not able to find one on that.
Okay. All right, great. Thanks very much for another great talk. Really appreciate it. And we have one more talk from Pearl today. And there is also a question from Shadman as well there showing up in the chat space. So Nidhi, if you could just answer that as well in the chat space. And Pearl, share your screen whenever you're ready.
It's saying that you disabled participant screen sharing.
Yeah, sure. Go for it now.
Okay.
Great, it's coming up. And we can hear.
I did.
And thanks for giving the last talk today, Pearl. Really appreciate it. And do
Yeah.
ask questions in the chat space. Thanks. You can full screen if you wish. That looks perfect.
Okay, sounds good. So the kinase that I chose for my presentation is WNK lysine deficient protein kinase 2, also known as WNK2. I'll start off with the structure. There are two domains located in WNK2. The kinase domain, which stretches from amino acid residues 195 to 453, and a coiled coil region, which spans from amino acids 1928 to 1959. The kinase domain is important for phosphorylating amino acid serine and threonine, and the coiled coil region is important for WNK2's protein-protein interactions. It has three homologous regions, which make its structure very similar to other WNKs in its family, but its function differs greatly from them. There are no signal peptide sequences in my enzyme as confirmed with Phobius. This makes sense due to the fact that it's involved in intracellular signaling pathways and found in the cytoplasm. SwissPalm confirmed that there were no palmitoylation sites. Myristolator confirmed that there were no myristoylation sites. And finally, by using PREPs, I was able to confirm that there were no prenylation sites. So expression and localization. WNK2 is a globular protein that is found in the cytosolic region of the cell. It is not membrane bound, and the confidence for this placement is very high, as confirmed with the deep TMH-MM prediction and with MODA. This enzyme is mainly located in the heart, brain, and skeletal muscle, but it also has some functions in the small intestines, liver, and colon. Okay, so the function and then biological significance. The main function of WNK2 is to act as a serine and threonine kinase. With this function, it will phosphorylate RAC1, which is an activator of PAC1. So it will render RAC1 inactive. Without RAC1, PAC will not be able to will not become active and won't phosphorylate MEC1 at serine 298, which is a key phosphorylation site for MEC1 to become active and activate ERK1 and 2. Ultimately, this means that there will be reduced activation of ERK, leading to a decrease in cell proliferation. Researchers were able to discover this by reducing the amount of WNK2 in certain cells. The results show that when WNK2 is reduced, or a kinase dead
Sorry. Okay, Pearl, your connection's a bit weak, but just keep on going.
Okay, do I go back a bit or no?
No, that's fine. Just keep on rolling. Unless you want to go back.
Okay.
A little tight on time, but I'll hang out for longer if necessary.
Okay, I'll just go back a bit. So researchers were able to discover this by reducing the amount of WNK2 in certain cells. The results showed that when WNK2 is reduced or a kinase dead mutant was overexpressed, it led to an increase in G1 and S progression in the cell cycle. And it also sensitized cells to be activated by low amounts of EGF. Multiple cancers show low expression of WNK2 and high activation of oncogenic signaling. Pathways which makes sense because WNK2 has been seen to inhibit cell proliferation, invasion, and migration of cancer cells. It is a tumor suppressor gene that is being tested to see how it can be used to target certain cancers. There is controversy in understanding whether or not WNK2 activates autophagy in cancer. In some cells, such as glioma, leukemia, and breast cancer, WNK2 has been seen to inhibit autophagy, whereas in other cells like colorectal cancer, expression of WNK2 induces reduced expression of autophagic markers. There's still a lot of research being done about WNK2 impact in the biomedical world. But as far as regulation goes, researchers were able to discover that when the promoter region of the protein is methylated, this leads to the silencing of its function. These are my references. Are there any questions?
Great. Are there any, there's a couple questions in the chat space. Maybe, can you see the chat space or shall I ask?
Um, let me. Yeah, I can see Tiffany's. So it says you mentioned WNK2 has a similar structure to its family members, but they still have different effects. What are those different effects? So that's a good question. So as I mentioned, WNK2 has been seen like as a tumor suppressor, and in the other three members of its family, they all have different mechanisms, but what they all have in common is they're seen as they have oncogenic roles in cancer progression. So there's like a difference between obviously WNK2 suppressing cancer and then those other family members promoting cancer.
Great. If people need to go, that's totally fine. I understand. But Pearl, if you can hang out for a minute and maybe answer one or two more questions, that'd be great.
Okay, sounds good.
Are there any drugs designed to target WNK2?
Well, I didn't find any drugs to target it, especially because it's kind of a cancer, it's like a tumor suppressor, so you really wouldn't want to have a drug to target it. But I did find that if you can regulate it by methylating it at its promoter region to silence its function. So I guess there could be a drug to promote that, but I haven't seen any drugs.
Great. And what are the functions of those homology regions?
I'm not sure. It didn't really mention that it had a function, just the fact that those homology regions are present in all of the other family members.
Okay, great. And could WNK2's role in phosphorylation of RAC1 have any impact on diseases beyond cancer, such as neurodegeneration or cardiovascular disorders?
Well, since RAC1 plays a role in cell movement and growth, if it's inactivated by WNK2, that could have implications on diseases like neurodegenerative disorders or cardiovascular diseases, where cell migration and survival are important.
Great, Pearl. Thanks so much for all of your answers there and everybody for presenting and having great questions. Really appreciate it. We got one more class of presentations coming up on Friday, and then I guess on Wednesday we have a guest speaker. So look forward to seeing you in class as usual in person on Wednesday. Thanks, everybody, and see you next time. If you have any questions, feel free to hang out. I'll stay here for a minute if anybody wants to chat. And thank you again for participating and giving great talks. A lot of effort went into these, obviously, and I really appreciate it. Okay, I'm going to say goodbye because no questions.
Sorry,
Oh.
I just have a quick question.
Yep, go ahead.
For citations for the slides, is there a specific citation we should use, or is do we just make sure there's some sort of citation at the end of the? That's what I've kind of seen from a lot of people, but I just wanted to ask.
Yeah, that's a good question. And you don't need to put citations. Usually I just put them on the bottom of the slide kind of innocuously rather than a separate slide for references, because at the end of the day, you're telling a story and people aren't going to click through during your slide presentation. It's just if they want to go back in the future and maybe check them. I will be sharing the video, and so people might want to check it out later. But I would say just focus on what you think the audience wants to see and hear during the talk, and the references, yeah, just put them in the bottom if they're relevant to the data that you've shown or the figure that you've shown on that slide. Okay.
Oh, okay, sounds good. Thank you so much.
Okay, thanks. Good question. Anything else? Yeah, please.
Hi, Professor. I had a question. My presentation is coming up next day. So I saw one of the participants actually mentioning the conflicting findings in the
Yep.
results. So I have a few conflicting findings in terms of transmembrane regions, like different softwares, they give me conflicting results. So would you like me to address that, or like how do I go about that?
Yeah, like I think all programs have slightly different algorithms and confidences possibly as well. So I would say, first of all, use your judgment to come up with what you think is, looking at all the evidence, the most likely to be accurate. Like if it's a transmembrane domain, look at the localization. Is it cytosolic or membrane bound? Do programs like Phobius and Filius agree? Well, TMHMM is even like higher, I think, confidence. So check that one out as well.
Okay,
Check the
yeah.
structure. If it's buried in the middle of a kinase, it's not going to be transmembrane. It's going to be a buried helix in the kinase structure. And so just take a look at all that data and then come up with what you think is the kind of obvious solution. If it's really not obvious based on your intelligence, then say, look, this is remains ambiguous because whatever. Two high quality methods are giving you different answers. Does that make sense?
Yeah, okay. And are there like a hard and fast rule regarding the number of slides? Does it have to be 5?
No, it's not hard. I asked people to stay to 5 minutes, like 4 minutes talk and 1 minute Q&A, just so we could stay in the schedule. But I mean, you can have as many as you wish, but the recommendation is about 5, because typically it takes at least a minute to talk through a slide. But I mean, if you have an animation or a couple slides where you just add panels, that's fine. So there's no specific requirement for 5 slides. That's up to you. That's just a guideline.
Okay, thank you. Yeah, I was thinking of putting the animation, if it works, in a separate slide. So yeah, I'll probably just do that.
Yeah, just try it beforehand with a colleague because we had a couple animations not work today. And I think Canva, maybe it's not quite as, working quite as well with a Zoom for some reason. So just try and do a beta test with one of your friends or even with yourself. You could probably just set a Zoom session and see if it works. All right.
Yeah, okay, I'll try that. Okay, thank you so much.
Okay, great. Have a great weekend. See you soon.
Sorry, just like a follow-up question, just a quick follow-up question. With regarding to like conflicting ideas, so that's what I was kind of doing. I'm kind of going through all the databases and seeing what I have and pooling together and comparing them all. So if there is something, because what I was thinking is there was some contradictions. So I was thinking of going into the literature for that. If there aren't matching databases, because even some of the databases they'll like randomly not even work. I have a lot of the screenshots for a lot of the databases, which is good, but so it'd just be a good idea to go to the literature if there are some conflicting.
Yeah. Yeah, like keep in mind you got 4 minutes to tell a story, and you can't take people down too many dark alleys. You want to tell them, the main thing you found, and maybe mention that like you had that there was one conflict, let's say, but I wouldn't bring up all the conflicts. The reality is every method has a false positive and a false negative rate. And so no method is perfect. So we know that. But using the time that you spent analyzing the predictions and also going through, if there's are any papers about that quandary, that's even stronger than a prediction usually, like assuming the data has been collected well and analyzed properly with controls and all that kind of stuff. I think the experimental data usually is going to weigh much heavier than the predictions on their own. Okay.
Okay, sounds good. Thank you so much.
Okay, thanks. Appreciate it. Any other questions? If not, see you on Wednesday. Okay,
See
bye-bye.
you. Bye.